are in environmental control. And importantly, you can't apply parametric release to aseptically filled products, only to terminally sterilised products. There's also a big section in the module on cleaning and disinfection, and this is vital to the maintenance of facilities and to ensure that contamination risks are minimised. Facilities and equipment need to be designed so that they are fit for the intended purpose. So they need to be cleanable. Clean rooms need to be designed in a way that they have smooth surfaces, no ridges and so on. And it's important to use the correct cleaning and disinfectant agents and to ensure they've been validated. So we've got time for another exercise. Go into your groups and just jot down what you think are the key elements of a good cleaning and disinfection program. Again, spend 5 to 10 minutes on the exercise, feedback, and I'll give you some thoughts in a few minutes. So if we can pause the video. Okay, so I'm back with you and we're going to have a look at cleaning and disinfection programs. So, some thoughts. Well, obviously the area needs to be cleaned and disinfected regularly, and you should have a written procedure and program. When carrying this out, you need to have a log, so you can demonstrate, particularly to regulators and for yourselves, that you cleaned when you said you cleaned. Sufficient time and resources should be dedicated to the cleaning activities, and certainly the cleaning and disinfection will vary according to the criticality of the area. If your cleaning and validation is delegated to a contract company, it's important to have a technical agreement in place. It's important that staff are trained in GMP, that their training is documented, and that you're following an SOP that describes the cleaning process. The cleaning agents used should be validated, and if you're operating within Europe, then they need to be validated to the Committee for Normalisation within Europe, the CEN. And you should have described how you prepare the cleaning agents, particularly for diluting down concentrated disinfectants. The cleaning procedure must take into account of time, temperature and mechanical action. You need to prove that you can remove dirt, so this is where you need to go with detergents first, followed by disinfectants. So disinfectants should only be applied to relatively clean surfaces. It's important if your surface is dirty, the disinfectant simply won't work. It's also important to note that disinfectants require a set contact time. So this is how long the disinfectant needs to remain in contact with the surface for. It's also important within Europe that disinfectants are rotated. Scientifically, this has little bearing, but it's a regulatory and GMP requirement that disinfectants are indeed rotated. Okay, so that's the end of the formal part of this course. And I'd now like just to wrap up with a few points about the exam and mention something about communication. So you'll see from the screen there that the exam is 2 hours long. And there are a series of essay questions. So this is the exam format. So what you'll need to do is to answer one question from a choice of 3, and the answers are to be in essay style. So the aim is to test theory and also to show the practical application. Now, a good revision technique is that you plan your answer for 10 minutes, then you write for 25 minutes, and then spend about 5 minutes checking your answers through. So that's just an idea there of exam technique. Okay, and then just a little bit about communication. You'll see from the screen there that I have my work email address, which is the first one, and my home email address, which is the second one, and there's also there details of the website which I spoke about at the beginning, to do with pharmaceutical microbiology, news, topic and debates and points. So it goes without saying, I'm sorry I haven't been able to be with you today, but I hope you found this interesting, and I'll be here on the Saturday to address one-to-one tutorials and so on. So I hope you found the exercise useful, and I hope this provided a good overview of the module if you've yet to sit it, or for exam preparation if you haven't sat it yet. So I hope you enjoyed the rest of the day, and wish you well with your studies. So goodbye, and hopefully see you soon.